It's 15 hours. I think we can start. So we are still on translation, and this time around, we want to look at the process of initiation in translation. So the understanding here is that we appreciate that the process of translation is taking place in the cytoplasm in both eukaryotes and in prokaryotes. And the process itself utilizes the information that is on the mRNA, and this information is utilized to make proteins. Now, we also understand at this point that the information on the mRNA is read using a language that in the previous lectures we looked at as a genetic code. In the genetic code, we all acknowledge that this is a 4-letter language that is a nucleotide bases. And when they're on the mRNA, they are read in threes, a sequence of 3 nucleotides, which we refer to as a codon. And we understand at this point that a codon is just a sequence of 3 nucleotides that can do 2 things for the cell. Either it can carry information for making an amino acid, that is to say the codon can be translated into an amino acid, or the codon can give information like a signal such as where to start from, like the start codon. That one, of course, we know the start codon gives us an amino acid methionine. But other signal codons just signal the machinery to say this is where you should stop. And these are referred to as stop codons. So what this means is that when you look at a codon, you don't always get amino acids. There are times when the codon itself will just give us signals such as to stop, which stop codons do not give us amino acids. So now, in this lecture, we want to understand what do we need for us to initiate translation. And again, at this point, we also appreciate the fact that the amino acids don't just come to the ribosome on their own. They're brought to the ribosome by the tRNA. We looked at the features of the tRNA. The amino acids have to be attached to the tRNA on the 3' end of the tRNA. We also appreciate that the tRNA also has an anticodon. An anticodon, again, is just a sequence of 3 nucleotides that has a corresponding complement of nucleotides to the codon on the mRNA. So with that understanding, and I hope you had the chance to look at some notes or just read so that as we start this lecture, we're on the same page. So today we want to look at the process of translation initiation. And in this process, what the machinery, in this case the ribosome and other factors that are involved need, is to recognize the start codon. And at this point, this start codon we know is on the mRNA. If you can see my cursor, the mRNA is this line, the red line running from the 5' end to the 3' end. So we're trying to say the ribosome has to recognize the start codon. In addition to that, the ribosome has to bring in, or should I say, when the ribosome recognizes the start codon, we need a tRNA to bring in the correct amino acid that corresponds to this amino acid. So now, in this figure, you see that in the structure of the ribosome, you've got this amino acid site, the S site, peptidyl site, and the exit site. This amino acid, the tRNA that is coming to bring in the amino acid, which corresponds to a start codon, is coming in the P site. Remember, in the previous lecture, we said that when these tRNAs come to interpret the information on the mRNA, they come in the S site. But the first tRNA that comes into the ribosome to recognize the start codon on the mRNA enters the ribosome at the P site. This tRNA, if you can see up there, it's designated as FMET. This is formyl methionine tRNA. And in the next slide, I'll explain why that is so. So the take-home message on this slide is the first thing that the ribosome needs to do is to know where does this information on the mRNA start from. And at this point, we know the codon that marks the beginning of the information on the mRNA is the start codon, AUG. AUG is the codon for methionine. The tRNA, therefore, that's going to come in the P site and is going to form complementary base pairing, the codon and anticodon, must carry methionine. One thing about this slide also is that this tRNA that is recognizing the first codon on the mRNA, the start codon, does not enter the A site. It enters the P site. And there's a reason why that is so, and I hope by the end of this lecture, we're able to appreciate that. Now, why does this first tRNA that brings in methionine recognize our start codon enter the P site? So on this slide, I want to draw your attention to this mRNA molecule. From the 5' end, running all the way to the 3' end. This mRNA molecule has got an AUG at this position. It also has an AUG at that position. So for this mRNA, this AUG is the start codon. What this means is that this is where the information that the mRNA is carrying should be read. The reading frame should start from here. This is where the information should be read from first. So then how does the machinery distinguish this AUG from that one? There must be a way to distinguish them. And think of it in this way. If you have a sentence and you start reading it halfway, definitely it's possible you're going to get the full instructions or the full information, but it is always best to read the sentence right from where it starts to the full stop. In the same way, this information has to be read right from the start codon all the way to the stop codon. What that means is that the cell must have a mechanism to distinguish this AUG, which marks the beginning of the information, from this AUG, which can be found anywhere else along the mRNA. To distinguish them, these cells have got two tRNAs that recognize the AUG codon for methionine. The tRNA that recognizes the AUG at the start codon is formulated, it's like modified. And this is why it is referred to as N-formyl methionine or just FMET. This tRNA, it also brings methionine to the ribosome, but its role is to recognize the AUG at the start of the mRNA. While the tRNA that recognizes an AUG similar to this one will also bring in methionine, but it is not modified. In this sense, it is not formulated. So this is one of the mechanisms in which a cell, or should I say the ribosome, can distinguish between an AUG, which marks the beginning of information on the mRNA, and one that is found internally along the mRNA. And also, the methionine, the FMET, the N-formyl methionine tRNA, that brings in the methionine in the ribosome, the one that reads the start codon, does not go into the A site. It goes straight into the P site. However, the tRNA that brings in methionine to recognize any other AUG enters the A site. What you see in this figure is a tRNA carrying methionine. This one recognizing any other AUG that is found along the mRNA. So here there's a difference. You've got N-formyl methionine FMET, and you just have a tRNA for methionine. Now, these two are different because even the way the positions they occupy in the ribosome recognize the start codon, goes straight to the P site. The tRNA that also brings in methionine to recognize the codon anywhere else along the mRNA enters the S site. And this allows the machinery to know where to start reading this information on the mRNA. So this is just something that you need to understand before we start the process of translation initiation. To understand also that the tRNA at this point is activated. What that means is that we already have the corresponding amino acid attached to the 3' end of the tRNA. So let's look at the components that we need for translation initiation. So we know that we need the molecule that's going to provide us the information, which is the mRNA. In addition to that, the mRNA is also providing us with a position where this information should start, which is a start codon. In addition to that, it's also going to provide us with information to say this is where this information stops. It's also going to have stop codons on it. In addition to that, we understand that this process of translation is taking place in the cytoplasm, but where in the cytoplasm? It takes place on the ribosome, isn't it? Both in prokaryotes and in eukaryotes. Keep in mind, the process of translation that we're discussing right now is in a prokaryotic system. That is why on top, on your title, we're saying bacteria. Now, on this slide, we're saying that we need the 30S ribosomal subunit. Now, here, this is for us to understand that the ribosome, before initiation starts, is not yet assembled. We have got the small subunit and the large subunit. So what normally happens is that at the start of translation, the small subunit needs to be independent of the large subunit. In this sense, if it's bacteria, the 30S ribosomal unit, which is a small subunit, must be independent of the large subunit, which is a 50S. Okay. In addition to that, we also need this initiator tRNA. In the previous slide, I said the initiator tRNA or FMET, this is the tRNA that is going to recognize the first codon, the AUG, that designates where this information on the tRNA should start. And this tRNA must go into the A site. In addition to that, initiation also need these initiation factors. These are factors that help sometimes to bring in the tRNA into the ribosome, into the A and the P site. Some of these initiation factors ensure that the small subunit does not prematurely, associate with the large subunit before the assembly is complete. We also need a source of energy. Remember, during amino acid activation, we used ATP. In initiation, we're going to be using GTP, which is guanosine triphosphate. So in a similar way, ATP provides energy. This process of initiation is also an energy consuming or dependent process which requires energy. And this time around, the energy is from GTP. Then also we need the large subunit, which is a 50S subunit. At some point when all the components of initiation have assembled on the 30S ribosomal unit, we also need the 50S to come in and complete the 70S ribosomal unit, because remember, the ribosome functions as a complete unit of the small subunit and the 50 subunit. But what we're trying to say here is that these subunits are assembled sequentially. So ultimately, the 50 subunit will come into place, associate with the small subunit, and collectively, we're going to have the 70S ribosomal subunit. We also need ions, magnesium ions. These ions stabilize the association of the ribosome with the mRNA and also the tRNA. So we're going to look at each one of them sequentially as we discuss initiation. So at this point, there's two slides from 1 to 7, should I say from point 1 to point 7, is just for us to understand that the process of translation initiation requires a number of components. Yes, at this point, they sound like there are a lot. In fact, it's even difficult for me to remember what number 1 was, what number 2 was. But I hope that as we go through the slides, you'll be able to understand each one of these components and what function they're playing in initiation. So the first thing that we're going to do is to assemble the ribosome. We understand the whole ribosome must be a 70S. That means it has got a 30S ribosomal unit. It also has a 50S ribosomal unit. So to form the 70S ribosomal unit, the first thing that's going to happen is the formation of a 30S ribosomal pre-initiation complex. So, like I said, the 70S ribosomal unit is not assembled, at once. It is assembled sequentially. So the first thing that happens is that the 30S small subunit of the ribosome forms a pre-initiation complex. And during this time, this small ribosomal unit is not interacting with the large subunit. And initiation factors, such as initiation factor 1 and 3, they help to stabilize the ribosome. You're going to see that IF1 is going to block the A site, while IF3 is going to stabilize the ribosome so that it does not bind to the 50S ribosomal unit. In addition to that, the small ribosomal unit also needs to position the AUG site, the start codon, in the P site. Remember, in the slide previously, I said that the first tRNA that's going to enter the ribosome goes into the P site. What this means is that when the ribosome binds to the mRNA, it must position the AUG site in the P site. How does it do that? So it does that with the help of this sequence that is referred to as the Shine-Dangano sequence. It helps to align the mRNA, in the P site. Of course, the unit of the small ribosomal unit that associates with this is a 16S RNA. Then, of course, there's recruitment of the initiator tRNA to come into the P site, and this one is facilitated by complex of initiation factor 2, complex of GTP, and also the ribosome. Sorry, and also the tRNA, and together they move into the P site. And this is why I said the first tRNA goes straight into the P site and not the A site. And this is because, and you're going to see very soon, the A site is blocked by one of these initiation factors, allowing for the first tRNA to go into the first empty site, which is the P site. And then finally, you're going to have, assembly of the 70S ribosomal unit, where now we bring in the 50S ribosomal unit. So these are just notes. At this point, I want us to go to the first step of forming this 30S. It's a 30S ribosomal pre-initiation complex. Okay, 30S means the S is a small ribosomal complex. S is a seed unit. Okay, but we know this is a smaller subunit of the ribosome. So the S is not like it's a small subunit and things like that. That's a unit of segmentation. So now, how do we assemble the 30S pre-initiation complex? I'll draw your attention to this figure. So what you see here in pink, if this is pink, even on your side, this is a small ribosomal unit. And what we see here is amino acid site. I'll just refer to it as an A site. And this is a peptidosite. This is where the peptide bond is formed. But you also know here there's also an exit site, okay, the E. But of interest right now, we are concentrating on the A site and the P site. So we said that the formation of the 30S pre-initiation complex, or indeed just to have translation initiation, we also need initiation factors. And these include IF3 and IF1. So what does IF1 do? IF1 comes in and blocks or associates with the A site of the small ribosomal unit. And once this initiation factor, IF1, is bound to this location, it is no longer free to associate with any other tRNA. And then IF3 comes in to bind to the small subunit, not in the P site, like what is shown here. I think I should have moved it. It will bind elsewhere. And the binding of IF3 ensures that the small subunit is stable enough and does not associate with the 50 S subunit. Why is this important? Because remember, at this point, we need the mRNA to come and slot itself into the ribosome. So if the 30S ribosomal unit associates very quickly with the 50S ribosomal unit before the mRNA is brought into the ribosome and positioned, it will be difficult for initiation to start. So to prevent premature association, IF3 comes in to stabilize the smaller subunit. The next thing that needs to happen is to recognize the start codon of the mRNA. Now, the mRNA itself has got a sequence that is referred to as a Shine-Dangano sequence. This sequence is found upstream of the AUG start codon. Remember, in our previous lectures, we've looked at how we designate a gene. So right now, when I say that the Shine-Dangano sequence is found upstream of the start codon, and we all know at this point, the start codon is AUG. When I'm saying upstream, meaning it is found this way. So the ribosome itself, it associates with the Shine-Dangano sequence to ensure that the AUG site on the mRNA is positioned in the P site. And we know why this is important, because at this point we understand the initiator tRNA or the FMET enters the ribosome at the P site. And if it's going to bring in the first methionine or amino acid to recognize this, we need for that methionine, for that tRNA to form a stable anticodon codon match. There should be no misalignment. So what we're saying here is that before assembly of the 50S ribosomal unit, certain things have to happen to the small ribosomal unit. What are those things? You block the A site to ensure no premature mRNA tRNA comes in. In addition to that, you stabilize the small ribosomal unit so that it does not associate with the large ribosomal unit. Why this is important? You want to bring in your mRNA and also ensure that the start codon is well positioned in the P site. The ribosome does not just guess that this is where the AUG is. It is helped by what is referred to as a Shine-Dangano sequence. This helps in the positioning of this AUG in the P site. One of the features of this Shine-Dangano sequence is that it is actually rich in purines, or should I say the G's and the C's. This is more on the Shine-Dangano sequence, and it's important for us to remember that. We're saying that the Shine-Dangano sequence is on the mRNA, and it associates with a small subunit of the ribosome. Okay? Now, the Shine-Dangano sequence itself, it is found upstream of your start codon. You can see this is a start codon, AUG. This Shine-Dangano sequence associates with the ribosome, facilitating the positioning of the AUG in the P site, and we know why that is important. Now, we know that in the P site, the first tRNA that's going to come in must be that one that's carrying methionine. But this AUG has been distinguished from all other AUGs that could be found on this mRNA because this tRNA that's going to enter the P site is formilated. In addition to that, this tRNA is going to enter the P site because the A site is blocked. So the take-home message from this slide is that the AUG is placed in the P site. It doesn't happen by accident. This is because the mRNA itself has a sequence that is referred to as a Shine-Dangano sequence found upstream of the AUG that first associates with the ribosome and ensures that the AUG is positioned in the P site. So now let's look at what happens when we have our AUG positioned in the P site. It has been aided by recognition of the Shine-Dangano sequence by your ribosome. So what we see here is our tRNA. This tRNA has to carry methionine. Remember, it is modified. That is why you see the FMET. If you see a tRNA with an F, meaning it's N-forming methionine, meaning it has been modified. This methionine tRNA recognizes the AUG at the start, meaning the one that's going to recognize another AUG is not going to be formilated. This is like a marker. Now, this methionine, this FMET, is not brought to the ribosome by itself. It forms a complex with initiation factor 2 and GTP. Remember, this GTP is going to provide energy at some point. We're going to see as we go on in initiation. Now, what you see here is you have IF1 in the A site, and that is why I said these initiation factors ensure that this tRNA fits into the P site because the A site is blocked. In addition to that, the small subunit at this point is not capable of, is not going to stably bind or associate with the largest subunit because of this instability caused by IF3. This allows for the mRNA to be well positioned in the P site as well as the incorporation of the FMET in the P site. Of course, these are notes for you to read and so on and so forth. Now, formation of the 70S ribosomal initiation complex. At this point, we already have our 30S initiation complex. You have your mRNA well positioned in the P site, that is the AUG, and also the A site is blocked. Now, to form the 70S ribosomal unit, we need to bring in the 50S ribosomal unit. So what normally happens is that the bringing in of this or the 50S ribosomal unit, that is association of the 50S ribosomal unit and also the 30S ribosomal unit, causes instability of some of these factors such as IF3 that were, preventing this small ribosomal unit from associating with it. And also the destabilization, or should I say, the release of these initiation factors is made possible by hydrolysis of GTP, providing energy for all those molecules to be pushed out of this ribosome. So what normally happens is that when you have your 50H ribosomal unit, all these factors that were blocking the S site must be released, and also any factor that was holding onto the ribosome, preventing it from associating with the 50S ribosomal unit, also has to be released. This process is in part facilitated by the help of energy from hydrolysis of GTP. So up to this point, what are we saying? We have not yet started the process of initiation. What we need to do is to assemble the ribosome. The ribosome itself is assembled sequentially. First of all, you need to stabilize your 30H ribosomal unit, the small subunit. Bring in your mRNA, ensure that the AUG site is well positioned in the P site. In addition to that, the S site of the ribosome must be blocked. And then bring in your larger subunit. Now, the binding of the large subunit to this smaller subunit can only be possible if these initiation factors such as IF3 and all the others are released. If they're not released, remember, the small subunit will not stably associate with the larger subunit. So up to this point, when you have your A site is empty because IF1 has been removed, your small subunit is associating stably with the large subunit because IF3 has been removed. And also, you have your initiated tRNA well positioned in your P site with aid of IF2, and it has also been released because we no longer need it at this point. What you have right now is referred to as the 70S ribosomal initiation complex. This is now complete because the ribosome now has its small subunit and its large subunit. In addition to that, it has got the mRNA that it's supposed to start reading in the correct reading frame. That is to say, the start codon is positioned in the P site. So that when I say the reading frame has been established, what I mean by that is, remember, these codons on the mRNA are read sequentially in 3. So what this means is that you have AUG. If it's going to be read into methionine, of course, because it's methionine, the next codon should also be read in 3. So meaning that the binding of the initiated tRNA to the start codon in the P site also establishes a reading frame to say this is where you should start. And when you start at this position, you're going to read this information sequentially in 3, or should I say codon by codon. These are not explaining everything else that leads to the formation of the 70S ribosomal subunit. Now we move on to elongation. Up to this point, we have assembled our 70S initiation complex. What is a 70S initiation complex? You have your large subunit and your small subunit associated, stably associated. You have your mRNA and the start codon well positioned in the P site, and you also have your initiated tRNA well positioned in the P site, forming a stable codon-anticodon interaction. And we know that interaction between the tRNA anticodon and the mRNA codon, they can only stable interact if there's complementary base pairing. What it means is that the N-forming methionine must be carrying methionine because the the AUG codon must be translated to methionine. So up to this point, how do we now add other amino acids? How do we read the next codon after the AUG? That is what elongation is all about. So the first thing you do is to initiate translation. Then the next thing is to elongate. Elongation in translation refers to the process at which now, from the start codon, we now need to start reading each codon on the mRNA codon by codon until we reach the stop codon, which is the full stop. So these are just notes for us to to look at, that is to say what is going to be happening in translation. We're saying this is a second stage of translation. In bacteria, this process first of all has to begin with a functional 70S initiation complex. And at this point, we understand what a functional 70S ribosomal initiation complex is, okay? And also, we understand that for this 70S ribosomal subunit to be functional, the S site must be free to receive a new tRNA that can read the codon in that position. So then, successful elongation requires the following components. You must first have the functional 70S ribosomal unit. You must also have a pool of amino acid tRNAs. What do we mean by that? At this point, we understand what this means. These are tRNAs that have been charged, meaning that they are carrying amino acids on the 3' end that are corresponding to the anticodon on the tRNA, and that will also correspond to the codon on the mRNA. That's what we mean by amino acid tRNAs. And then also, we also need elongation factors. Remember, for initiation, you had initiation factors. To elongate, you also need elongation factors. Just like initiation factors, they also have different roles that they play. And as we go on sequentially, we're going to see what these initiation factors are. I think I even put up a summary of what these elongation factors are. So for example, elongation factor TU, the TU just stands for thermal and stable. Okay, not that you need to know what any of this is just for an interest, but what's important is for us to understand that elongation is an enzyme dependent process. It also requires energy. It also needs other factors, initiation factors, that come in to help the process to be conducted in a successful way and also at appreciable rate. So, for example, I was trying to say elongation factor TU, it delivers amino acid tRNAs to the A site of the ribosome. Elongation factor TS, it regenerates the GTP. After the energy has been utilized in the process of elongation, the GTP has to regenerate it so that it can be reused, because remember I said this process is an energy dependent, it consumes energy. And then we also have elongation factor G. This is a translocase. We're saying that the ribosome from the start codon has to start reading each codon sequentially. Now the ribosome, what that means is that it has to move on the mRNA. So if right now the ribosome from our previous slide is at AUG, for it to translate, sorry, so it's at AUG. For it to translate the next codon, for example, if it was ACG, it has to move to that codon. So to help this ribosome to move to the next codon, the mRNA, it utilizes elongation factor G. Of course, this is also a process that requires energy. So now, this is just an overview of elongation. I'm not too sure whether I should use this slide to explain, or maybe I should go to the next one. Let me use this one. So the first step in elongation is that the S site is going to have a codon, and this codon has to be translated. What it means is that this S site needs to receive an amino acid tRNA, meaning a tRNA that is carrying an amino acid that corresponds to this codon on the S site. So now, how is this tRNA, which is carrying the amino acid that corresponds to this codon, brought to the ribosome of the A site? And I want you to pay attention. At this point, we know that in the P site, coming from the function of 70S ribosomal subunit, we already have our initiated tRNA sitting with the methionine. So we want to read the next codon. At this point, we know the A site is now free from those initiation factors that were sitting there. So at this point, a tRNA can come into this space. The tRNA that's going to come into this space must carry the correct amino acid on the tRNA A site 3' end that corresponds to that codon. How does it come there? So again, this is through the aid of initiation, elongation factors, as well as energy. So in this case, we're going to have a complex of elongation factor TU GTP and the tRNA that is carrying the correct amino acid. This is going to be brought into this A site. What I'm trying to say here is that this tRNA does not come to the A site on its own. It is kind of escorted by these elongation factors that form a complex with GTP, and together they come into the A site. At this point, the tRNA in the P site is independent, carrying its own amino acid, methionine. And at this point, you also have another tRNA that has come into the A site, also carrying its own independent amino acid. The two amino acids are not yet linked. At this point, we just have the P site carrying a tRNA with an amino acid and the A site carrying a tRNA with an amino acid. Now, when this tRNA forms a stable codon-anticodon interaction with the mRNA, that is to say, the codon on the mRNA corresponds to the anticodon on the tRNA, and that can only happen if the tRNA is carrying the correct amino acids. The GTP is hydrolyzed, releasing the GTP and the elongation factor from the ribosome. At this point, it's important for us to understand that this GTP has to be regenerated because it's going to be required again. Because we're trying to say that these tRNAs that are reading the mRNA or that are brought into the A site are not coming there on their own. They need to be escorted by elongation factor, TU, as well as GTP, forming a complex with that tRNA. Once there is stable codon-anticodon base pairing, and we know these are the complementary base pairing rules, the GTP EFTU destabilizes and leaves the ribosome, and it has to be regenerated so that it can be used again. So it's like a cycle. Okay, so regeneration of this elongation factor GDP complex is facilitated by elongation factor TS. That is why I said these elongation factors, they serve different roles. For example, in this case, we understand that the tRNA is not coming to the ribosome on its own to bring the amino acid. We can say at this point, for lack of a better term, it is escorted. Who is escorting this tRNA to come into the P site? It is a complex of elongation factor TU and GTP forming a complex with the tRNA. So it's going to be elongation factor TU, GTP, tRNA, whatever amino acid is going to be carrying. And together they're going to come into the A site. The moment that tRNA forms a complementary association with the codon, that is to say the anticodon on the tRNA and the codon on the mRNA, on the stable binding, these elongation factors dissociate. And also there's GTP hydrolysis. Now what has remained here is GTP. So this one is regenerated back to GTP, okay, by this elongation factor TS. This is very important for us to understand, and I hope when you go back on your own, you're going to realize it's a very simple process. You must first understand your P site is blocked because already the AUG site was placed there with its own tRNA. The A site is now free. Now, the tRNA that's going to come there is the tRNA carrying the exact information, corresponding information to its codon, meaning there's going to be an anticodon coding match. Once that is done, the elongation factor that escorted this tRNA to the ribosome has to dissociate with the aid of GTP hydrolysis. Now, when you hydrolyze your GTP to GDP, it has to be regenerated. This is facilitated by another elongation factor, TS. So the next slide, these are just notes for us to go through that process. I want us to look at the next step, which is now the peptide bond formation. Remember, I said up to this point, if I draw your attention to this figure, you have your AUG positioned in the P site. Here, the exit site is empty, so you have your AUG positioned in the P site. You also have your formyl methionine, the F-met, carrying methionine, which corresponds to this AUG codon. In the previous slide, we also understand that another tRNA amino acylated tRNA, that means it's carrying an amino acid, has also been brought in the ribosome, this time around in the S site. Between these two amino acids, they are not linked. At this point, they are independent. Okay. Now, for us to make amino acids, we know that, or should I say, amino acids are linked by peptide bonds, meaning that the amino acid in the P site must form a peptide bond with amino acid in the S site. The understanding here is that it can only happen if these tRNAs that are holding these amino acids have formed proper stable anticodon-codon matches. When this is happened, like I said, for GTP to hydrolyze and let go of that elongation factor TU, is because there was complementary base pairing between the codon and the anticodon on the tRNA. So what normally happens is that the amino acid in the P site is released to form a peptide bond with the amino acid in the S site. Normally what happens is that the amino acid in the S site utilizes its amino end to attack the carboxyl end of the amino acid in the P site. And this amino acid is released from the P site completely to the amino acid in the S site. What you see here, for example, is that the formylmethionine that came in first in the P site, it is no longer carrying the methionine. That methionine has been transferred to the amino acid to the tRNA amino acid in the S site through the formation of this peptide bond. This reaction is catalyzed by peptidyl transferase. Now, here, what are we saying? You have two amino acids, one positioned by tRNA in the P site, the other amino acid positioned in the S site. Up to this point, they're independent, but we know to make proteins, these amino acids must be linked by peptide bonds. The peptide bond is formed between the amino acid of the end terminal of the amino acid in the S site and the carboxyl end of the amino acid in the P site. So meaning that this amino acid is going to attack that amino acid, leading to this amino acid being transferred completely to the S site. So what we see in the S site is an extension of a dipeptide because they're just two at this point. You've got the one that was in the N terminal and in the P site, and now we also have the amino acid that is already sitting in the A site. What has happened here to this amino acid, to this tRNA, is that it is no longer holding on to any amino acid. And when this happens, this tRNA is no longer forming or should I say, is no longer stable in its conformation, because remember, what makes it stably bind to this codon is that it is carrying its amino acid. The moment the amino acid is no longer on that tRNA, it becomes unstable despite the complementary base pairing. So what normally happens is that there's a shift. Okay, this ribosome is going to shift one codon going this way. I'll explain later because I also want us to understand that one, here we're trying to say that the first amino acid that is in the P site is going to go to the amino acid in the S site. When that happens, the tRNA is no longer charged. Charged tRNAs do not stably bind to the mRNA in the ribosome, so they kind of bind loosely. This allows also for the ribosome to shift. Of course, we know the shifting of the ribosome requires energy and, of course, elongation factor G. Now, in the next slide, I want us to also look at translocation of the ribosome. Which direction is it moving? How is a ribosome reading the mRNA? At this point, we understand what is happening in the S site, we understand what's happening in the P site, and also in the exit site. This is where the tRNAs that have lost or donated their amino acids exit from. It's like an exit door. I don't know if you have a house, there are those houses where you have got the main entrance and the back entrance. So probably this one should be That's, the exit door of your house. So think of it in this case. So now I want us to understand one thing. How is this ribosome reading the mRNA? Is it 5 to 3? That is to say, it is moving from the 5' end of the ribosome, going to the 3' end of the ribosome, that's towards the 3' end of the ribosome, or it is moving from the 3' end of the ribosome, going to the 5' end. Well, with what you can see, if I draw attention to this figure, the ribosome is reading the mRNA. Here the mRNA is shown in green, starting from the 5' end, meaning that the ribosome starts from the 5' end, reading the mRNA sequentially codon by codon towards the 3' end. So what it means is that we were, from the previous slide, the amino acid that was in the P site was given to the amino acid that was in the S site, or should I say there was a peptide bond that was formed. Now, the tRNA that remained in the P site no longer had the amino acid, and because of that, it becomes unstable and exits the ribosome by moving to the P site. What you see here is that when the ribosome shifts one codon going this direction, this tRNA that was holding on to those amino acids is going to move in the P site. What I'm trying to say here is that, remember, this tRNA was initially in the S site. But then at this point, we need to read the next codon after it. So meaning the ribosome has to shift 3 nucleotides or just one codon towards the 3' end. When that happens, it means there'll be a shift in position. The amino acid, the tRNA that was what was holding on to the 2 amino acid is going to move to the P site. The tRNA that was in the P site that has now lost its amino acid will move to the exit site, and now another codon will be positioned in the A site, which will not have a tRNA. So now, because the A site is again empty, but it has an mRNA with a codon, another tRNA can come into the A site and form complementary base pairing with this codon. When there is a match between the codon and the anticodon of the tRNA, that is to say, the tRNA is carrying the correct amino acid. Thanks. It's going to form
Home Alone 2 director Chris Columbus described the delicate process of getting
a peptide bond being formed between the amine group and the carboxyl group. Now, when that happens, this polypeptide chain will be transferred again to the amino acid in the tRNA in the S site, at the S site, leaving this tRNA again without any amino acid. And there'll be a shift. So it's like the sequence has to be repeated over and over again. It's important for us to understand that the translocation of the ribosome, that is to say, when the ribosome has read a particular codon, for it to move to read the next one, this process requires GTP hydrolysis and also elongation factor G. So this is just an illustration of what is going on there. You have the ribosome has translocated meaning it is moving from the 5' end of the ribosome, moving towards the 3' end of the ribosome, moving codon by codon. Once the amino acid that has been translated, so what should I say? Once the tRNA moves from the A site to the P site, another codon moves into the A site, and it also needs to be read. Because the A site is empty, another tRNA carrying the corresponding amino acid, that is to say, that can be translated for this codon, is brought into the ribosome in the A site. And again, there will be a peptide bond that will be formed between this polypeptide chain at this point carrying 2 amino acids with the amino acid in the tRNA in the A site. And there will be a transfer of this dipeptide to this amino acid. And again, there will be translocation. And the process will keep on repeating itself. Now, this slide, I just wanted us to picture what is happening every time we're saying that the polypeptide, so the mRNA will keep on being read from the 5 to 3. But then what is the direction of your protein? Remember, we also said proteins have got some form of orientation. They have got an N terminal. This is where you find the amine group. And they also have the C terminal where you find the carboxyl group. So what you see in this figure is the tRNA that is in the P site, and this tRNA is in the A site. Just imagine that this is a ribosome. We're saying that the tRNA is carrying an amino acid in the P site, and another tRNA is carrying an amino acid in the A site. Now, the amine group of the amino acid of the tRNA in the A site is going to attack or react with the carboxyl group of the amino group of the amino acid in the P site. So now what you're going to have when the peptide bond is formed is that we're going to still maintain the amine group. You can see this is the amine group and this is a carboxyl group. The amine group is from the first amino acid that was in the P site, and the carboxyl group that you're seeing here is a carboxyl group coming from the amino acid of the tRNA that was sitting in the A site. And you're going to sequentially add more amino acids coming from this end. Ultimately, you're going to have a protein with an orientation reading from the N terminal to the C terminal. But the ribosome that was reading this information was moving from the 5' end to the 3' end. So you have this information coming all the way from the way the DNA is read, the way the ribosome, the way you read, the way we decode information from the DNA. It is in the 5 to 3. The direction which you're reading this information also has an implication on the product that is formed. We can clearly see that because of the ribosome reading this mRNA from the 5 to 3, even the protein that is formed also has a specific orientation. It is from the N terminal to the C terminal. Termination. Now, like I said, the mRNA is not just carrying information for us to make proteins or amino acids. No, it is also carrying signals. At the beginning, we know the first signal that we've encountered is the AUG. So at this point, we also know that the mRNA also has signals that also show that this is where the information should stop, like a full stop. And these signals are referred to as stop codons. These codons, you must know them. You must also know the start codon. The stop codon in this regard is an important signal that is required in translation termination. That is to end termination, because they must show the protein machinery, or in this case, the ribosome, those elongation factors to say you should no longer regenerate your GDP. I think at this point, the information has stopped because you don't want to be spending energy anyhow. So now, the signals on the mRNA that help the protein machinery to know where the information stops are these stop signals, and you've got 3 of them: UAA, UAG, and UGA. Now, it's important for us to also understand that because these stop signals do not code for any amino acid, they will not have a tRNA. What you're trying to say here is that if you are a stop codon, you should not have, what would the tRNA bring to the ribosome? Because remember, the stop codon just means a full stop. It's not meaning anything. It's different from the start codon. It's also a signal, but at least the start codon gives us a methionine, meaning that it's going to have a tRNA bringing that methionine to the S site of the ribosome. So meaning that for termination, we need what are referred to as release factors. These release factors kind of mimic what the tRNAs do. Okay, they come into the S site, but they kind of like, mislead the ribosome to think that they are also carrying an amino acid. But when it's time to transfer that amino acid from the P site to the amino acid on the A site, there's nothing. And ultimately, you have the release of the polypeptide. So the process of termination was saying that is facilitated by intrinsic signals that are found on the mRNA. These signals are also read in codons, and there are 3 of them. And for each mRNA, it will only carry one, because remember, there's no way one mRNA is going to carry more than one stop codon. You can think of it, if you're going to write a sentence and you put full stops inside the sentence, and yet you want it to be one whole complete sentence, the information won't read correctly. In a similar way, think of a stop codon as a full stop. You can only have it at the end of a sentence when that information is written in its completeness. So what we're trying to say here is that these release factors, they're the ones that come in to recognize the stop codon on the mRNA, and what they do is they trick the machinery to think they're also carrying an amino acid. But when it's time to transfer that amino acid to the polypeptide chain, the amino acid in the A side, there's nothing there, and you have your polypeptide chain released. Up to this point, guys, these are notes for you to go and read extra. This is just another slide that I wanted us to look at, just to appreciate the fact that in prokaryotes, we said that transcription and translation both take place in the cytoplasm. Okay, of course, we know that the genetic instructions in bacteria, they're found in the nuclear region. But because of the proximity to the cytoplasm and because there's no membrane-bound structure, there's no wall around the nuclear region, this information, the process of transcription, is connected to translation. What this means is that when the mRNA is being transcribed, that is to say, the information from the DNA is being processed to make mRNA, which we refer to transcription. The process of translation is also taking place. What this means is that on one end, you've got transcription taking place, on one end, you've got translation, because the ribosomes can bind to the mRNA and start synthesizing the protein. And all of them, as you can see, they all move in the 5 to 3 prime direction. So now you can imagine if one of them was moving the opposite direction. So all this, when you look at even just how this information is read sequentially, you appreciate why the direction of transcription, translation, in a way, are in the same direction. It facilitates for efficiency, because while this guy is being transcribed, it can also be translated. What you have here is just a summary of everything that you need. And this is the end of translation. What are some of the take-home messages? Translation just converts the genetic information into a protein. And it's important for us to understand. In translation, you're not converting all the information that is found in the nucleus, that's the DNA, no. You're only getting a segment of DNA, which we refer to as gene. So what happens in translation is that the information that was the segment of DNA we're referring to as a gene was first of all taken into mRNA and finally into a protein. Initiation establishes the reading frame. When you have the formation of the 30S pre-initiation complex and the 70S, what you need to understand there is that before the ribosome can assemble as a whole, you need to position the mRNA to ensure that the reading frame, that is to know where the information starts, is well identified. And then elongation extends the polypeptide, a very simple process that requires energy and also elongation factors, initiation factors to come on board. And finally, termination is achieved by release factors. And this is because the stop codons do not carry any amino acids on them. So the next topic, we're going to look at gene expression in prokaryotes. And I'm going to end here. That's going to be tomorrow. Up to this point, thank you very much for attending. And I've sent the notes. Yeah, I think I sent the notes to Mike. I think you'll be able to share. So tomorrow we start a new topic. And if you are having challenges with understanding this topic, it will be good for us to, it will be good for you to let me know, because on my end, I'll take it that everyone else has understood. But I'd appreciate, go back home, read it on your own, and then you can tell me, I don't understand this area, help me understand. And I can even have a makeup lecture for those of you who feel you're struggling to understand before we move on to other. Well, of course, we're going to move on, but I can accommodate some who might be having challenges understanding translation and transcription. Up to now, guys, enjoy the rest of the day and the evening ahead.
Madam.
Bye for now.
Madam. Madam. Not working.
Mr. Jacob.
Yes, boy. What's your question?
A wonderful lecture, and your notes are always very precise and easy to read. But I just have a general question concerning the complexity of how these topics, like how are they make these observations? The labels, the well-connected observations, were these all under a microscope or?
I don't think I understand the question very well.
Or is I was just genuinely curious as to how these scientists were coming up with these observations and well-connected explanations.
Okay, so for example, for us to understand the genetic code, remember there is, I don't know whether you can go and read about Sanger sequencing, okay? How the nucleotides at some point were labeled, you know, and how they found out that the DNA itself has got, the combination of those letters. And also when you think of the ribosome right now, even if I took a plant cell, you've been to the lab, I'm sure you've looked at the plant cell. None of us have seen the plants, the ribosomes, isn't it? But when you use electron microscopy, probably you're able to see those things. So most of this information has come about because of technology. And even today, some of the things that we thought were correct are not actually correct because there's new technology coming up, powerful machinery coming up. So more and more information is being discovered. So much of this, you might have to go and, read about discoveries of how even DNA was found to contain those nucleotides and things like that. Read about electron microscopy, chromatography, and combined all those techniques. It wasn't just one discovery, no.
Thank you very much.
Anything else, guys? I think up to this point, enjoy the rest of the day.